It's going to be a special night tonight. I want to welcome Tim Kamp back to Wednesday night at the lab.
Okay, thanks so much, Tom. Can everybody hear me all right?
Great.
So it's really a great pleasure to be here and a lot of fun to help celebrate 20 years since the discovery of human embryonic stem cells. And I have to tell you, I thought this would be a lot of fun to put together this talk. 20 years didn't seem like that much. It turns out I've been here those whole 20 years. I came here in 1996 and have fortunately been able to be involved in some of this research and see how it unfolds. And as I started to gather materials and think about everything I was going to tell you, I realized that we only had an hour and not a few weeks, and so I had to pare it down. And the only way I really could figure out how to pare it down a little bit is to try not to bore you too much with my own story of how I've been involved in this research and gone through some of these discoveries and participated in some and really seen the field explode over the time since that first discovery 20 years ago. So what I'll try and cover today are, I'll give you a few definitions so we're all on the same page, although I know this is a very well-educated audience, so I probably don't have to spend much time on definitions. Talk about the discoveries and the progress we're making, how we're getting the cells we need, what we're doing with them in the lab. You can't really talk about this without touching some on the ethical questions, the public policy that's impacted it, and ultimately a little political influence as well. Talk a bit about patents and startups that have arisen from this, and then where are we with clinical trials. So I think that should fill the hour, all right? So let me just put this into perspective with a little timeline here and how things really are expanding pretty rapidly. We really didn't have much of an idea or a concept of stem cells really until the early 1960s, when blood stem cells were first described in some detail by Till and McCulloch as a stem cell in your bone marrow that's able to repopulate your blood and keep your blood system going. And that sort of gave concept to the idea that there are master cells in the body that stay in a primitive state and then can grow into the specialized cell types and continue to replenish those cells throughout time. But for today's talk, the big discovery, one of them leading up to it, was the isolation of mouse embryonic stem cells. These were the first embryonic stem cells isolated, and two different groups published it simultaneously in 1981. And they found that if you took inner cell mass cells from a mouse blastocyst, so the early embryo in a mouse, that under the right conditions they could grow those in the lab in a tissue culture dish, and then they could put those back in another mouse and make it in a pseudo-pregnant mouse and make a new mouse. So they maintained their master stemness, just like the fertilized early embryo. So that was really a breakthrough, but it wasn't so easy. Once these mouse embryonic stem cells came along, it wasn't so easy to get them from other species. It seemed pretty straightforward. If you could figure out how to do this in a mouse and get those cells out of the mouse, grow them in tissue culture, and then you have a master stem cell in your lab, you should be able to do it for any species. But it wasn't easy. It wasn't until 1995 that Jamie Thompson here really made the less heralded but probably more important discovery, and that was how to isolate embryonic stem cells from a monkey, from rhesus, because that gave him the conditions that would allow him a couple years later to isolate human embryonic stem cells. November 6, 1998 was the day it was published in Science. Shortly before that, Dolly the sheep was created by somatic cell nuclear transfer, or a cloned sheep. So a lot happening in that space. In 2006, induced pluripotent stem cells that we'll talk about a little later were first isolated from the mouse. Again, the mouse leads. And then soon thereafter, Jamie's lab here at UW and Yamanaka and colleagues in Japan isolated human iPS cells. And we've gone on to see now some clinical trials with ES cells, iPS cells, And now there are nuclear transfer ES cells, like Dolly the sheep, another form of ES cells over the years. So things are changing pretty quickly here in recent years, but that gives you a little timeline perspective. And so we're going to obviously focus from 1998 on. So just to be sure everybody understands what an embryonic stem cell is, they are derived from the inner cell mass of the pre-implantation blastocyst. That simply means after the sperm and egg join together to fertilize and form the zygote, which starts to divide and then forms an early cystic structure with very simple and primitive cells in there called the inner cell mass, that if you're careful and have the microsurgical techniques, you can take out these inner cell mass cells. And if you figure out the right culture conditions, and initially it was on feeder layers of cells, that you can grow these cells in culture. The amazing thing about embryonic stem cells is that they self-renew indefinitely in culture. That means they just keep growing and growing. As long as you take care of them, you can keep getting more and more of these embryonic stem cells, which is really an amazing feature. Pluripotent, they can give rise to all cell types in the body. If you're a developmental biologist, there are 3 primary germ layers that form our bodies, and they can form all the cell types in there. So these are master stem cells, and they're clonal. That means one embryonic stem cell can grow up and can form any cell type in your body. So those are the special features and the definition of embryonic stem cells. And so the breakthrough that we're celebrating this year was the isolation of human embryonic stem cells in 1998 by Jamie Thompson from, in this case, surplus in vitro fertilization embryos. And what that means is patients who have fertility problems, who have the sperm and egg combined together in a culture dish in the laboratory to do in vitro fertilization. Subsequently, if there are embryos that they're not going to use that would either be cryopreserved or discarded, they were donated for research. And from those donated surplus in vitro fertilization embryos, Jamie was able to isolate these embryonic stem cells. These lines are still growing today and are still widely used around the world, as you'll come to see. This is also credited with really empowering the concept of regenerative medicine and opening our minds to the fact that maybe if we can use stem cells or better understand stem cells, we can figure out how to regenerate our bodies, regardless of what the particular problem is. If we can get regeneration or new parts, that would be a good thing. Maybe it's using stem cells. Maybe we figure out from stem cells how to exploit the endogenous repair mechanisms in our body. So that's the big concept behind regenerative medicine, but I think Jamie's discovery really pushed that concept to the forefront and got people thinking about the potential there. But also I think the human embryonic stem cells are probably pretty useful in other ways. But what good are they in a dish? Well, they give us human cells that we can study in a dish, and that's something we didn't really have before this. Because, for example, if you wanted to study brain cells or heart cells, it's hard to get living heart cells, living brain cells out of patients and keep them alive in a dish that we can do our research studies on. Human embryonic stem cells gave us a way to form these heart cells or brain cells in a dish that we can then use for research. It could also be for testing drugs or safety testing in drugs, or maybe we want to study models of disease. Do something to the cardiomyocytes or the neurons or the pancreatic beta cells in the dish and study how that can mimic a disease. Why does it matter to have human cells? We've got animal cells, but why is this a big deal? And again, as Tom mentioned, I'm a clinician as well. I'm a cardiologist, and when I came here to UW, I was interested primarily in this sort of thing, electricity in the heart. I still am, for that matter. So you're going to have to bear with me because a lot of the descriptions are going to have something to do with electricity in the heart. And as a cardiologist, I recognize this ECG, the PQRST, And it's due to the underlying action potential or electrical signal in the ventricles. And the reason I bring this up is I show you a human action potential just to compare it to a mouse action potential. They're pretty different looking. And what does that mean? That means different ion channels, different proteins are contributing to that action potential. And if you want to figure out how the human heart works, electrically anyway, it's kind of hard from the mouse heart compared to the human heart. And this isn't even comparing for the time scale. This is occurring about 10 times faster in the mouse heart. So it's one of the reasons we think these human stem cell-derived cardiomyocytes, in this case, might be useful. How do we get them? Cardiomyocytes, if that's what you want to study, well, just like normal development, as embryos develop and the heart forms, in utero, if we do it in a dish, it also takes multiple steps and multiple intermediate stages, which I'm not going to bore you with, but we know many of the different genes that are turned on and off. We have names for some of these different cell stages and various markers to sort them out. But suffice it to say, it's a bit of a road to get from this master stem cell to a cardiomyocyte or to a neuron or to a skeletal muscle cell, whatever cell type you might be interested in. There is a developmental or differentiation pathway that we have to follow. So we first did this with our ability to form cardiomyocytes using something called embryoid bodies. Now, an embryoid body is basically when you take the pluripotent stem cells out of the specialized culture conditions they are and put them in media with serum, and you allow them to differentiate. And when we did this, if you have good eyes, you can see something moving there. Right there, it moves every once in a while. Or it was anyway. Now it's gotten tired. I'll see if I can wake it up. And so it started moving again. And so that was our first clue that these cells could form heart muscle cells. Unlike most of the cells, when we look under a microscope in our tissue culture dish, they don't usually move, right? So here were some cells contracting in a dish, which was a pretty remarkable finding for those of us who were fortunate to first be able to see this around 1999 here. And that hint of cardiomyocytes got us to move forward. This is the embryoid body, and just a little corner of it is actually contracting, but it's proof of principle that they can form cardiomyocytes. Turns out there are different types of cardiomyocytes in the heart that I show schematically here. And you know that. The lower chambers are ventricles, the upper chambers are atria. They have different jobs. They develop different pressures in the body. There's also pacemaker cells, nodal cells. So not surprisingly, they all have different electrical signals or action potentials. Of course, I bring that up because I want to show you that when we took those stem cell-derived cardiomyocytes from the embryonic stem cells, we could see different electrical signals consistent with different types of heart cells in the dish. So that was encouraging that actually the stem cells could form different types of cardiomyocytes that had electrical signatures that experts could recognize as heart-like electrical signals. And furthermore, they look like human heart-like electrical signals. So that's good news. These could be useful. But we were frustrated. I showed you just that little corner of the embryoid body was contracting. So most of the cells in there weren't heart muscle cells. And if you're in business to try and study heart muscle, that's kind of annoying. Of course, if you're in business to study the other cells, it's not so bad. But we wanted the heart cells. And so we had to try and figure out better ways to do this. And here's one way we tried with Sean Pelichak, who is in chemical and biological engineering here. And he's an engineer, and he knows how to make things. So he made some microwells that are basically to the non-engineer, little muffin pans. And so this, what you see here is where cells are growing in a little muffin holder. And you can see the side view cut out here. So here is the holder, and here are the cells growing in there. The idea is when you empty out the muffin pan, and you can control how big that is, you can make different size embryoid bodies. And that's what we did, made different size, so we controlled it instead of just allowing the embryoid bodies to form more spontaneously. So we could make embryoid bodies of different sizes and then see which size worked best. And we found that embryoid body of a certain dimension, about 300 microns, work best to make cardiomyocytes. So that was a step forward, but it was only one of many ways as this field expanded, and that's one of the other things to highlight. We started out doing this research. There weren't many studies being done around the world because it all literally started right here in Madison, not far from here in the UW hospital where the cells were isolated, and my lab next door in the old hospital where we did some of the work, and then the Y-Cell Research Institute. And so over time, though, others got involved and figured out other ways to make embryoid bodies besides the microwells. They used patterning and centrifugation, and then started adding growth factors to try and get better at it. And then others said, well, let's put in some other cell types to sort of encourage these cells to differentiate into cardiomyocytes. And yet other approaches were, let's not make those embryoid body clusters at all. Let's grow the cells as single cells, as a monolayer of cells, so one layer, and then be smart and add the right growth factors and signaling molecules so we can turn this monolayer into contracting cardiomyocytes. It turns out that proved to be a pretty effective way to move forward, and now we can get it's not going to play that one. Okay. So if you blink quickly, you can see that whole thing moving. So we can now get whole plates with millions of cardiomyocytes contracting. Sorry that this one isn't showing that contraction pattern. But now, instead of just having a corner of the embryoid body, we get the whole plate of cells contracting. And so we, and now many labs around the world, can do this. And it's true for cardiomyocytes, but the improvement in techniques and methods to get the cells into the particular cell lineage you want or cell type you want, such as beta cells for the pancreas, skeletal muscle, a variety of neurons, all those techniques have improved dramatically over the years and allowed people to get large numbers of these cells from embryonic stem cells. So just here at UW-Madison, the ability to differentiate these cells into a variety of different cell types has been demonstrated by many, making them into neurons. Suchun Zhang's lab has been very successful, and you've seen some impressive images over the years from his lab. You can also get a variety of different blood cells, Igor Slupkin's group. You can get retinal cells, and Dave Gam here at UW and his lab has been responsible for a lot of that work. Astrocytes, which are supporting cells in the brain. Masa Suzuki has been successful in making skeletal myotubes. And this just gives you an idea of some of the different cell types that have been differentiated here at UW, but this is just the tip of the iceberg. Around the world, these have been duplicated and many other cell types have been differentiated. But I can't really talk about this anniversary without touching a little bit on the ethical questions and stem cells. This is something we had to all address. As researchers, we had to think about this, and as a society, we had to think about this. And the derivation of human embryonic stem cells does require the destruction of a human blastocyst or embryo. There's no ambiguity about it. And so that makes people ask, is that equivalent to destroying a human life? And that's where you'll find differences of opinion. And obviously, if I'm here talking about this, I don't think it's the same human life as you or I, or I wouldn't be doing this research. But certainly other people have different opinions. Also, somatic cell nuclear transfer, Dolly the sheep, reproductive cloning, brings some real questions out. Even exploiting oocyte donors so you can get the best oocytes for reproductive cloning or for therapeutic cloning. Should we worry about creating chimeric animals when we transplant human cells into an animal, putting human neurons into a mouse brain? Is that a problem? Or putting human stem cells that get into the gametes of a monkey? what happens with reproduction. So human iPS cells may help with some of these things because they don't destroy an embryo, but as technology gets better, what if those cells actually are able to be used for in vitro fertilization? So I think the questions are not easy, and they continue to be present, but we continue to work through them and think about them. It has impacted me as I got this birthday card not long after I started doing some of this research. I'm still not sure if it was a joke or a question, but you can take it for whatever you want. So obviously there are some potential concerns here. So this led to a variety of thoughtful projects to sort through some of this. The U.S. National Academy of Science put out guidelines for human embryonic stem cell research in 2005. Alta Sharo here was on that committee, as was Norm Foss, two of our bioethicists here, who helped craft some of those guidelines. It asked for the formation of escrow committees at institutions, embryonic stem cell research oversight committees, and we have one here at Wisconsin that reviews all the research using these cells to try and make sure they're used in the most ethically appropriate fashion. The International Society for Stem Cell Research also put out guidelines, and in the United Kingdom, the Human Fertilization and Embryology Authority provided a variety of guidelines as well. But this was a little complicated because the Bush administration was struggling with these newly isolated embryonic stem cells. Although they were isolated in 1998, by the time any real serious decisions could be made, George W. Bush was in office, and they came up with the compromise policy, in part thanks to our former governor, Tommy Thompson, who was Secretary of Health and Human Services, to come up with this idea that federal funds will be set aside to explore the promise and potential of stem cell research. And they want to use federal dollars to help attract the best and brightest scientists, ensure that the new discoveries are widely shared and directed to the public good, but they won't support any further destruction of embryos for ES cell research. So you've got to use what's out there as of August 9th, 2001. So they drew the line in the sand. At that point in time, that seemed like a pretty good deal because all the stem cell lines we had were there. But of course, as time wore on, that seemed less of an appealing solution because, like everything, we thought newer lines with new techniques would be better. So it allowed stem cell research to move forward in this country with federally funded dollars. No restriction on private research, but most of the research in this country is powered by federal dollars. So that ultimately led to the NIH to think, well, how can we get these stem cell lines out to people in high quality? And so one of the things that they did is the National Institutes of Health put out a call for a national stem cell bank. A bank that would hold all these human embryonic stem cell lines, all the ones that had been derived before August of 2001, and grow them up, make master banks, and then distribute them in a cost-effective way. Here at UW, we competed for that proposal, and it was awarded. The National Stem Cell Bank was here at YCell, led by Derek High and Jamie Thompson. And basically, Jamie's lines were easy to get here. They were already here. But that list of 21 lines that was approved by the Bush administration involved lines from all around the world. So much of the National Stem Cell Bank's job was to get those lines into the U.S. and expand them, make sure all the intellectual property was appropriate, and allow investigators access to that. So complicated business, which lines to use and how the NIH got them into researchers' hands. Just to let you know, that bank was funded from 2005 to 2010, and then the federal government didn't think they needed to do that anymore. And so what happened to that bank is the Y-Cell Institute here in town took over, and it became the Y-Cell Stem Cell Bank. It still has all those 21 NIH approved lines, but also a variety of other lines that are available as suggested here. It's gotten complicated because not all states are created equal, you might know, and some states had different ideas about this human embryonic stem cell research. And so the state that had the strongest idea, or the most advertising perhaps, was California. And California in 2006 passed Proposition 71. Proposition 71 basically gave $300 million per year for 10 years of taxpayer dollars to support stem cell research, basically embryonic stem cell research, because they thought the U.S. Federal budget was not adequately supporting a very promising area of research that could impact the health of their citizens, and by the way, they thought it might be good for their biotechnology industry. And so they made a major investment in stem cell research. Other states took different approaches. At this time, there were 6 states that outlawed human embryonic stem cell research, made it against the law in those states. And other states, New York, Connecticut, funded stem cell research with state money, similar to California, but to a much, much smaller scale. Did that matter? Probably did, because here's an interesting follow-up slide. So that was in 2006. Here's a slide that Tennille Ludwig from Y-Cell shared with me. And this is, where did those original 5 embryonic stem cell lines go? And how many went there? Well, a lot went to California, not surprising. A lot went to the East Coast, to Massachusetts, New York, and so forth. Less so here in the Dakotas, obviously a hot spot in the Midwest here, but somewhat reflecting the states that sort of encouraged or discouraged this research. So you can see it's a little complicated how this gets through the public policy and where the research is done. But in order to foster it here, we didn't get an infusion of state money here in Wisconsin, but we thought it was important to work together to make this research move forward as best we could. So we formed a Stem Cell and Regenerative Medicine Center in 2007. I've been fortunate to be one of the directors or co-directors since its initial founding. And now we have more than 100 PIs, or principal investigators leading labs in the Stem Cell Center, as well as a few hundred trainees. We have 5 different focus groups in the Stem Cell and Regenerative Medicine Center here, from cardiovascular regeneration, Amish Raval, Wanju Lee leads musculoskeletal regeneration, Anita Bhattacharya, the neural regeneration group, Emery Bresnik, the blood research program, and Sean Palachuk, the bioengineering group. We sponsor a variety of symposia and meetings, both locally and a little further. We support pilot grants and also a variety of training opportunities. Jordanic provided some of the materials when you came in. So this is our effort to keep our stem cell research here at the University of Wisconsin moving forward in the strongest way possible. Just to continue and get us up to date with where policy is now, the Obama administration changed the policy of the Bush administration soon after they took over in 2009. In March 2009, March 9th, this executive order came out entitled Removing Barriers to Responsible Scientific Research Involving Human Stem Cells. Here is the signing ceremony with President Obama, and this is a poor quality photograph. I apologize because I was fortunate enough to be there, but I'm not a very good photographer, as were several other UW stem cell researchers to celebrate this change in policy. And now there are more than 400 human embryonic stem cell lines approved for research by NIH funding. So this has opened the avenues up again. Okay, let's get back to science. Can you do that shift? We're going to get back to science now after that little detour, but we'll take more detours. I don't know who likes which better. So we're going to get back here to the iPS cells. And so this was really a landmark discovery, how to get induced pluripotent stem cells. What are induced pluripotent stem cells? Well, not to bore you with developmental biology, but it's pretty easy to conceptualize. We think of a master stem cell as at the top of the hill here. This is something that Waddington first conceptualized and drew. It's called the Waddington landscape. And then as development occurs, the ball rolls down to various valleys, getting deeper and deeper until it gets into the specific cell type it's going to be, whether it's a blood cell, a B cell, a macrophage, another type of blood cell, muscle cell. But it's sort of rolling down to become that specialized cell type. Thought to be a one-way pathway. But what induced pluripotent stem cells are is that now finding out ways that you can take that fibroblast or muscle cell and redirect it so it goes back to the master stem cell phenotype, so it can get its youthful sort of biology and become like an embryonic stem cell. So it really made our whole concept of developmental biology turn upside down, that we can do that. It was a fundamental and shift in the biology, and one that people were very skeptical of, especially after the mouse iPS cell discovery. But then Jamie Thompson here and Shinya Yamanaka succeeded in doing it with human material, in that they took skin samples, foreskin samples or other skin samples from donors, and then they transfected those cells with genes that are expressed at high levels in embryonic stem cells, pluripotency factors, and they overexpressed those for a number of weeks, and then they found the cells could grow indefinitely. They could self-renew in a dish, and they were pluripotent, just like embryonic stem cells. So by learning about embryonic stem cells, that's what gave us the idea and opportunity to get iPS cells. It taught us the factors that were important in making that cell type, that are controlling its DNA and its biology, and it also taught us the way that we have to grow the cells. And so without discovering embryonic stem cells for humans, we could have never gotten these iPS cells for humans. So it's a step down the road from that discovery 20 years ago. And this was 9 years after that discovery. So of course, the first thing we wanted to do is see, well, they sound good, but can they make heart cells or cardiomyocytes? And you can tell by the little squiggles shown here that this is how I identify a heart cell. It's by its electrical signal or action potential, and yes, looks like they can make those heart cells. And if we use antibodies to look at the contractile proteins, we can see these nice stripe patterns and colorful cells that we know are forming the myofilaments, or the things that make the heart cells contract. So that was exciting to see that from induced pluripotent stem cells, we could get cardiomyocytes too. But one of the real powers of induced pluripotent stem cells is that now I can take a skin sample or a blood sample from anybody in the room and make a master stem cell, an iPS cell, that's genetically matched to you. And then I can turn it into a heart cell. So I can have a dish of beating heart cells that are just like your heart cells, except maybe a little younger. The other thing that enables, though, is that those patient-specific cells can be taken from patients with diseases. So we can do disease modeling. So in other words, if you have an inherited disease, and in this case I'm showing you an example of a patient with inherited arrhythmia syndrome called long QT syndrome. So those patients have bad heart rhythms that can be life-threatening, and we want to understand what's happening at the cell level. What is the electrical signal at the cell level? And you can see, in contrast to those earlier electrical signals I was showing you, or a normal one here, that they have a much longer action potential, and it's doing crazy stuff here. Those are called early after depolarizations. And the craziness can lead to a bad heart rhythm that is life-threatening. So this allows us to study that in a dish and ideally develop new treatments. It also, as time went on, gave us the opportunity to make more complicated tissues. So I was showing you just single cells in a dish. With Carter Ralph here at UW and Toy DeLange, what they've been able to do is take these iPS cardiac cells and engineer whole pieces of heart tissue, not just a single cell. And they do that by putting them in these special culture dishes where the cells will fill in these troughs, and then they grow up and start to look like heart muscle down here. And if you look at it in a variety of ways, this is an electron micrograph, and if you're an expert, you can see evidence for sarcomeres, the basic contractile unit. And then they make these little tissues of contracting cardiac muscle. And this won't play either. Oh, doggone. Well, I checked the first movie as sort of my due diligence, but I guess I should have checked them all. But these little pieces of heart muscle, you can actually see them moving and contracting. This is much larger than the single cells. This is a couple centimeters long. So now we're getting pieces of tissue that are really big enough to see and to do something useful with, hopefully. But this also was being carried out in multiple other fields here at UW. And just to highlight a few, for example, Dave Gamm's lab has done beautiful work making actually little retinal organoids, so little parts of the eye. There's also been work by Eric Schuster's lab making blood-brain barrier cells, so the type of endothelial cells and supporting cells that protect your brain. More on the retina. Wanju Lee has made bone and cartilage in a dish, making stem cells into various forms of blood and blood vessels, even neural organoids by Jamie's group. And some of these blood vessels here, you can't see it move too well, but these are little microspheres going through engineered blood vessels. So the complexity and the research is moving forward with these stem cell products. Nowhere close to making whole organs, but getting more complex tissue constructs. And some of this has gone to make little tissues on a chip that may be useful, and there are a variety of projects here at UW funded by a number of different mechanisms, the NIH microphysiologic systems, the Environmental Protection Agency. They want to know about toxins in the environment, and they want to know what they do to human tissues. So if we can make these tissues on a chip that we can study these toxins, that would be very powerful. So one of the things that came to mind early on with the embryonic stem cell cardiomyocytes, in collaboration with my colleague Craig January, is that Craig had done a lot of work looking at the QT interval, which is a measure on the electrocardiogram of how long it takes for the heart to turn off after each heartbeat. And if it takes longer, if that QT interval is longer, that can be a problem. That can be reflected by prolongation of the underlying action potential in the heart, forming something called an early after depolarization. It can be induced by a drug, in that case called E4031, and it can lead to this heart rhythm here. So this is actually a patient that was at our hospital here in Madison at UW, now a few years ago, but she came in with a long QT interval, and she developed this rhythm. So it's fast, and it's ugly, and this is her blood pressure when that happens. It's not good. It doesn't show the bumps anymore. So the blood pressure goes down, and if this wouldn't have spontaneously broken, she would have been in trouble. This can be a lethal arrhythmia. It can also be self-limiting, as in this example. But ultimately, it turned out, due to detective work by Craig January, that a metabolite of this antihistamine that she had taken, estemazole, or norostemazole, had caused this side effect, had caused this life-threatening heart rhythm. This antihistamine subsequently was withdrawn from the market, and this, as well as several other discoveries about drugs inadvertently causing the QT interval to prolong, led to a whole new effort on the part of the Food and Drug Administration here in the US and other drug regulatory agencies around the world to try and look for this potential side effect in non-cardiac drugs. This is an antihistamine. You're taking it for your seasonal allergies, as some of you may be taking antihistamines like me right now, but hopefully not Hismanol. And so this is a pretty bad side effect if you're taking it for the sniffles. And so you want to figure out how to screen for this. So we thought these stem cell-derived cardiomyocytes might be good for doing this. And they might be useful at other steps in the drug discovery and developmental process. And we also thought the cells might be better in prediction compared to existing models. Could be less costly because many of the safety things that we're developing involve animals and increase the efficiency of testing and also allow us to sample a wide range of genetic variants. So this sort of concept suggested to us that perhaps this is something that we need to get into industry, and led to some patents and so forth. And ultimately, it led to a startup company forming to apply the technology. Cellular Dynamics was founded in 2004 with Jamie, myself, and Craig, and the Palais brothers, who were the business folks that helped, Bob and Tom. Tom is also a UW faculty member. And ultimately, that CDI was sold to Fujifilm in 2014, now has more than 100 employees in Research Park, and does make a number of different cell products from the pluripotent stem cells. This is just an example I'm familiar with because obviously I was involved, but there are many similar stories with other investigators around. So the first product from CDI were icell cardiomyocytes. They were red in color because of the way they designed the reporter to isolate the cells, so they were relatively pure. And they were frozen, so they could be shipped. And so you could get a lot of cardiomyocytes because CDI got very good at making those. And there have been many other companies that have taken advantage of the pluripotent stem cell biology in a variety of ways around the Madison area with a variety of startups and other companies, Promega, not a startup, but using the technology. So I think you're seeing that a lot of the discoveries in the university have gotten out into industry and hopefully are being useful. What about patents as a metric for what's getting out there? Well, here's a slide from Weissel and Worf. Again, Tennille shared this slide. These are the patents around the world that have used pluripotent stem cell technology. And I think you'll appreciate there have been quite a few, a lot in the US. Europe does some double counting because EU and single countries, they both can patent. So it's probably not quite that many more in Europe, but there are patents all around the world. So let's get back to science. We took another little detour. Let's get back to science. So what about cell therapy? Maybe that's why some of you are here. What are these things really good for? Can they really help human health? And so, again, I'm not going to tell you about eye disease because I don't have any expertise there, but I will tell you about the heart a little bit. And so for the heart, people have thought really not long after Jamie's discovery that, well, stem cells sound pretty good. If your heart is damaged, such as after a heart attack, and we know you lose a lot of functional heart muscle, we should put a stem cell in there and grow new heart muscle. Pretty appealing concept. And basically, this cartoon shows that some of the different cell populations that have been tried. And you can see that it hasn't been limited simply to one sort of stem cell. People have tried all sorts of different cell preparations, from fat, from bone marrow, from the blood. Embryonic stem cells have been used, the iPS cells, even stem cells putatively from the heart itself. And so that work is ongoing. Can the ES cell cardiomyocytes survive in the heart? And here's one of the major publications, not from here, but Chuck Murray's group at the University of Washington, where they put in 1 billion, 1 times 10 to the 9th, cardiomyocytes into a monkey heart. And these yellow cells are those human cells in a monkey heart. It's cut in the short axis, so it looks like a donut. And there's a large graft of these cells. Only problem is there's this rhythm that these cells are inducing. That's called ventricular tachycardia. That's one of those bad rhythms again that can be life-threatening. Fortunately, the rhythms lasted only a couple of weeks before they dissipated. But in this preclinical animal model, a monkey with human cells, we get some evidence that you can get new heart muscle in an injured heart. Is it going to be safe? What about arrhythmias? Many different reasons to worry about arrhythmias that I'm not going to bore you with the details. But if you make the heart better, if you heal the heart in the long term, it may actually be anti-arrhythmic. So that we don't know. It could work out all right. Another safety issue that we're concerned about are tumorigenicity. Can it form tumors? Obviously, we don't want that. Many of the genes used to reprogram the cells are potentially carcinogenic, and so we have to worry about those genes being turned off. Ideally, we have to worry about maintaining the cells in culture. They can change over time in culture and acquire traits that make them want to grow out of control. And we have to understand how best to test the cell preparations to be sure that tumors aren't really going to occur. What about immune rejection? This is a big challenge because with iPS cells, potentially we can do autologous, self-to-self. We can take cells from you, grow them up in the lab, and make the cell type and put them back. That turns out to be, with current technology, a pretty challenging road because of the time involved, the months literally to make the cells and the cell products. And with that time and resources are the cost. And so most of the effort is going towards allogeneic, so not perfectly matched cells. And so that would mean your body potentially can recognize those as foreign. So how can we get around that? One of the ways to try and study that that's being developed here in Will Burlingham's lab by a talented postdoc, Matt Brown, and Petros Aganapostopoulos, who's one of our pediatric surgeons, basically has this clever model where they take an immunocompromised mouse and take cord blood from a baby to get bone marrow stem cells, basically to repopulate the bone marrow. And then they take a little piece of thymus when that baby who has congenital heart disease undergoes surgery, and that's important to educate the blood cells, and basically get a humanized immune system in a mouse that we can then test our cell products in that humanized mouse. So it's a complicated mouse model with a lot of details I won't bore you with, but it gives us a way, short of putting the cells in people, right? We don't want to find out they're aggressively rejected when we put them into people initially. We need to study some of this in an animal model, and this gives us a way of looking at the human immune system in a mouse model. We're still trying to understand how these cells impact the heart. There are many ways. We'd like it if they just regenerated and repaired the heart, but they also secrete factors that could be beneficial and a number of other things that could help the heart. As a part of our effort to do this, we're also partnering with colleagues from University of Alabama, Birmingham, and Duke as part of the National Heart, Lung, and Blood Institute Progenitor Cell Consortium, and the goal there is to make large tissue patches. Oh, good, this movie moved. So, so you can see now that ruler up there, that's 6 centimeters. So we're getting to the stage where we can make big contracting patches of heart muscle, and here it is on a pig heart, and so trying to repair a pig heart. And so the technology continues to get better and better. It's gotten to the point now that the first in-man trial of human embryonic stem cell cardiac progenitor cells, so the cells right before they become cardiomyocytes, has been done and published by Philippe Menachet in Paris. It was just published this year, where they put those cells in a patch at the time of bypass surgery. So here are the cells in a patch, and sewing that on at the time of bypass surgery to see if that can be done safely. And so far, we know it's safe. There haven't been problems associated with it. We don't really know if it's effective yet, and obviously that's an important question. What about clinical trials in other areas? Right now, there are 5 areas that I'm aware of from clinicaltrials.gov where there are ongoing trials using human pluripotent stem cells for macular degeneration and other degenerative eye diseases. Some trials here in the US and in Japan for Parkinson's disease, some in Australia, China, and there's word there'll be one launched here in the US before long. Spinal cord injury was actually the first one to be done by Asteris here in the US in 2010, where they started enrolling and putting in a supporting astrocyte-like cell in the spinal cord to help it repair. We talked a little bit about heart failure and diabetes. ViaCyte is doing a trial with pancreatic beta-like cells in patients with juvenile onset diabetes. So the clinical trials are just starting with these human embryonic stem cells. Now, 20 years later, I can't tell you if they're going to help at this point. Certainly, I'm optimistic that there will be signs over time, but we're still on the steep part of the learning curve, and there's still going to be bumps in the road. Right now, for the trials that have been published, the safety signals have been good, so that means bad things haven't happened, but as clinical trials are designed, you start with safety studies that are small numbers of patients to make sure nothing terrible is happening. And then you can expand the trial to do enough patients to see if they're really effective in treating the problem. So what's happened with these original 5 human embryonic stem cell lines and their distribution by Y cell? Well, it's pretty amazing, really. So this little discovery 20 years ago, and as Tom said, Jamie presented at this lectern. He didn't have any idea what would happen. None of us did. But to think that now cells have been distributed to 45 countries, a total of 5,000, or closer to 5,400 now, distributions around the world, 6 continents. We're hoping for Antarctica, but they haven't asked for any yet. So these really have gotten everywhere. They really have changed the world. And the research is growing. That's what I said at the beginning, and I'll stick by it. It's growing. We can see that the number of publications, there was that one in 1998, but every year since then, more and more publications using human pluripotent stem cells. So this really has initiated an explosion in research and the beginnings of an explosion in regenerative medicine, and I think it's just the beginning. The first 20 years are hard, getting our feet on the ground and trying to understand what these beasts are and how best to use them. But I think the next 20 years you'll see even more remarkable success. So with that, I'll acknowledge that I can't acknowledge everybody that's been noted in this talk. There are just too many people. I have to thank the many people in my lab and my many collaborators, of which this is a very short list of. And I thank you for your attention and happy to take questions. Yes.
I noticed when I looked a couple of years ago, maybe a year ago, at WARF, I noticed that WARF had split between what I knew would be WARF, and now they have 2 separate funding sources, one for stem cells, and there's another one to support the colleges. How does WARF deal with stem cells now?
Well, I don't think WARF is really. I am not a representative of WARF, so I will only give you my impression. It's not official. But my impression is that WRF hasn't split in any way. WRF funds various initiatives. They fund the Morgridge Research Institute that's across the street. They fund a number of initiatives, and one of the initiatives they fund is the Y-Cell Institute. The Y-Cell Institute is a not-for-profit support organization to support stem cell research at the University of Wisconsin, and as its primary mission, and secondarily to help, as you saw, distribute these cell lines around the world. And so, so WRF does support Y-Cell, so some funds go there, although Y-Cell, because of its ability to charge for some of these products, is, does generate some revenue, so it's not just a sink for WRF money. So it's really not a division of stem cell patents and other patents. It's just an avenue that WRF decided they wanted to fund. Obviously, they think growing stem cell research will get them more patents, but it's not a separate division. Other questions? Tricks or treats?
A couple back here.
Okay, sorry, didn't see
I
back
wonder
there.
how extensive three-dimensional Petri dishes are in stem cell research?
Yeah, there's a lot more interest in three-dimensional culture. So we traditionally, with cell culture, have done it as monolayers. So you have a dish and the cells grow on the bottom of the dish. But now, as we think more and more about it and get better at it and want to make more tissue-like structures, no surprise, we're three-dimensional. Cells in tissues like to be three-dimensional. So there are advantages to doing that. There are many different formats to do that in, and certainly they are more costly than a simple Petri dish, but the devil's in the details. So some are very costly, some less so, and you can do homemade sort of with PDMS and a variety of other things you can do in the lab to make three-dimensional structures. And that, I showed you one thing with those not contracting. This is a little gadget that there's a little well in there where you pour in the cells, and actually there's a vacuum that pulls down that. So the cells stay in the well, this little line here, and then after a day you release the vacuum and that comes up, and they're released and are stuck between these two tabs here for a little linear strand that you see down here. So this plate is significantly more expensive than a standard tissue culture plate, but not out of the realm of reason, and they can be reused. That answer your question? Okay. Yes.
Do the cells need, I guess, food and oxygen or to survive, and how do you continually feed them and keep them from dying?
Yeah, that's the work that we have to do in the lab. Absolutely. The cells in the lab, just like your cells, need to be fed and need the nutrients and vitamins and other essential metabolites to keep going, as well as oxygen in the appropriate concentration and CO2 in the appropriate concentration. And so that means the cells growing every day or a couple of days, we have to suck out the media, the bath they're growing in, and put in fresh media with all those goodies in it. And we grow them in an incubator that is closed to the environment and has its own environment, which is at body temperature for human cells, 37 degrees, and has 5% CO2, which is like the CO2 we have, 21% oxygen. And so, although people play with those gas concentrations to some extent, but you control the environment they're in. You have to do that. You have to have the right gas environment so they can have oxygen and also release the CO2, and also you have to have the fresh media. Yes?
Can you comment further on the impact of the California law, how it's affecting it now?
Yeah, that's an interesting question. So that California law was in 2006 for 10 years and $300 million a year. So $3 billion, a lot of money. And so they have dispersed most of that money. They are trying to re-up the kitty to get the taxpayers to go in for more. But to my knowledge, that really hasn't made it to a ballot initiative yet. They don't want it to get voted down, so they're still trying to build up the support. So right now, they're sort of living on fumes from what they had initially. But what that did is it basically acted as a very strong magnet for stem cell researchers around the country and the world to go to California at the beginning of that 10 years, because all of us to run our research lab, we need some money. And so we have to write research grants to get that funded. California gave you another opportunity for research grants and to fund your research. So it gave another pot of money, which attracted many people because they could do more research. We always seem to have more ideas than we have money. And so that was appealing and got a big community of stem cell researchers in California, and also a variety of startup companies engaged in this sort of technology, and a lot of clinics as well. So that'll be the legacy of CIRM. It really did jumpstart stem cell research in California, which I think will have a long-term impact because just mass action. They've got a lot of research going on there now. They can compete with the rest of us now. We're all competing for NIH funding, but they got some pretty good folks in California as a result of that. Yes?
Are there any trials that you're aware of in Wisconsin for using stem cells on knees and hips replacements?
Not clinical trials. I am not aware of clinical trials using stem cells on knees and hips. There are some clinical operations that are delivering stem cell products to patients with orthopedic problems, but those, to my knowledge, are not part of a clinical trial. And a little skepticism about those clinics because they don't seem to be affiliated with larger clinical entities or research operations. So it's one of the dark sides of stem cell research right now is that there's a lot of for-profit use of unproven therapies popping up in the U.S. that people need to be cautious of. And so you really, if your insurance company tells you they're not going to pay for it and they want 20 grand from your pocket, you've got to think twice about that.
Do you think the nature of IRBs is driving research into the public or the dark sectors you said?
Well, I... Yes.
Repeat that question.
So the question is, are IRBs driving this research into the dark side? For those of you who may not know what an IRB is, it's an institutional review board or the Human Subjects Committee. It's the entity that clinical research has to go through to be approved that it's ethical, safe for patients. Obviously a good idea. We want to make sure that we do the safest clinical trials and patients make informed decisions. So it looks closely at the consent process. Do you agree with this and the risks and potential benefits? And so IRBs are certainly important. I think the genesis of the question is that sometimes the details and the complexity of getting a trial through an IRB can be a little overwhelming. Consent form, our consent form for getting iPS cells from patients in 2008 was 38 pages long.
Do you think if we had more citizens and fewer university people on the IRBs, it would move better?
You know, I think a lot of I can't blame it on the local IRBs. A lot of this is in response to the federal mandates and all the things that are required for federally funded trials, and so any university, part of their trials are going to be federally funded, so they have to stay to those standards. And the standards have gotten more and more complex, not out of malice or bad intents, but You know, it's just.
So, yeah, it happens. There's an awful lot of this dark stuff you're talking about that's moving offshore. And there's a lot of welcoming places. Is the United States likely to go backwater?
No. So to get back to your question, is the IRB inhibiting this research and making it go elsewhere? What's happening elsewhere, and the elsewheres in stem cell therapy, isn't really research, in my opinion, in many of these things. It's basically putting cells in and hoping for the best. Because why not? Stem cells, you got back pain, stem cells could be good for you. You got, Parkinson's disease, why not? Stem cells could be good. What do you have to lose? And the problem is you do have something to lose, because there can be life-threatening side effects, infections, and so forth. And so I think the research is ongoing. It just takes time, and it'll continue here. As far as going offshore, less of a problem now. There used to be the problem of stem cell tourism for some of these unproven therapies. So people would go to various places to get cell therapy. Vietnam, Haiti, that had less regulations. But that's not so much of a problem now, because you can get your stem cells for some of these questionable indications here in Wisconsin or at clinics in many states. There are now estimated 2,000 clinics doing unproven stem cell therapies in the United States for profit. So this is a problem.
Is it a good problem or a bad problem?
It's a bad problem, because you can ask the unfortunate women who are now blind in Florida because they got an injection of stem cells inappropriately in their eyes for retinal problems. We got some more.
I just wanted to ask you for since you're in a very similar field, when you first became aware of the thing going on at Harvard, that guy at Harvard who just...
Oh, yeah.
You must have been watching this, sort of going, what is happening?
Yeah, so what she's mentioning is there's been in the press, the New York Times a couple weeks ago, and then the Washington Post, there was evidence of falsification of data for stem cell applications for heart research, and it wasn't pluripotent stem cells, it was an adult stem cell source by an investigator named Piero Anversa, who is at Harvard. Harvard requested the retraction of 31 papers from his group, and also the NIH was paid back $10 million from Harvard for the grant money that they had given to Harvard for this researcher's investigation. It's,
Did you doubt you must have seen these as they were coming out, the journal articles?
Yeah, so this is a researcher who's been doing heart research for the last 35 years and publishing in the most prominent journals, so any heart researcher knows this individual. A lot of his research has been controversial, and some of his research really since the early 2000s has been questioned and rebutted in several publications. So there's been skepticism, but there's also been advocates. Not uncommon in science, right? People debate different things. But several of his key findings had already largely been discredited in most investigators' mind. What is new now is that finally that the universities and the NIH and so forth have done their investigations, and now there's no doubt there was fabrication of data and much was falsified. And it's a disaster for the field because so much time, energy. What's really bad is it got a lot of researchers going down that avenue, which basically is a blind avenue, wasted a lot of people's time. It also launched clinical trials, one of which you probably saw in the paper has been halted now because the basis for that clinical trial were the animal studies that his group did. So no two ways about it. It's a disaster.
Why is it a blind alley? What is that? Why? You say his approach was a blind alley, but why
Well,
technically?
without getting into too much of the details, he isolated a cell population which he said was a stem cell population that could grow new heart muscle using a particular way to identify the cells called c-KIT. It turns out that just wasn't true. It was false. But you can pull out cells from the blood or from the heart that are c-KIT positive that have that marker, and you can do various things with them. But if you're doing that because you think they're going to grow new heart muscle, you're deceived. And you've been deceived by very prominent publications and presentations that say these are, miraculously powerful cells. So that's why it's a blind alley. You think they're going to have a certain behavior, but they really don't because that data was falsified.
Is there a difference in quality or in characteristics between those embryonic stem cells that are derived from a fertilized egg, the original lines, versus those that came later from taking, let's say, some blood cells and reversing them back to the original stem cells? Are they exactly identically the same, or are they different in other ways? In ways?
That is an active area of debate. Some experts believe they're pretty similar. Others say there are potentially important differences. Certainly, the original embryonic stem cells have a better chance of maintaining the integrity of their genome than the reprogrammed cells, depending on how old those cells were when they were reprogrammed. Because as cells get older and older in our body, they tend to accumulate mutations, slight changes in the DNA. And so some people argue that the embryonic stem cells may be a little better in that way. But so far, functionally, they seem to behave very similarly. And if you do detailed what's called epigenetic analysis, looking at how their DNA is controlled so genes can turn on and off, there may be some subtle differences, but with the best new ways to reprogram cells, those are pretty much going away. The first iPS cells, like everything, the first generation wasn't quite optimal. The vectors were still there, but now we have what we call footprint-free reprogramming, so there's no leftovers in there from reprogramming. So the cells are better and better and closer, but there are likely going to be some subtle differences. And that's why the clinical trials that we talked about are primarily using embryonic stem cells. They've been around the longest, and there's less safety concerns. I think there was one here first, yes.
You've been working with cardiomyocytes. Has there been any work specifically on the electrical signaling nodes in the heart as well, for folks who have signal problems?
So perhaps what you're referring to is what we call a biological pacemaker. So that means cells that can act as pacemakers. And there are researchers who are working on that, and we can isolate pacemaker-like cells from these embryonic stem cell derivatives. And there have been proof of principle experiments putting some of these into a pig animal with what we call heart block that can pace the heart. And so that is an avenue of research. The good news about a biological pacemaker is it doesn't need batteries, theoretically never has to be replaced. The bad news is it's a little tricky to get it in there in a robust, reliable fashion relative to what we currently have available. So the electronic pacemakers that we can get off the shelf that the companies have made have gotten better and better. They basically are small computers with the electronics, very small, some wires, yes, but they're very, very reliable. And so most people aren't focusing on biopacemakers because the other therapy is so doggone good. Over time, I suspect that'll come, but it's not very far yet.
So are there any heritable aspects to working with stem cells, either intentionally or unintentionally introducing stem cells into germ lines and traits that are heritable?
Explain what you mean exactly. So putting human stem cells into the germline?
Suppose you felt that you could develop a superior CM cell, and would there be any, and there's a genetic basis to that, would there be any rationale, do you think, of somebody necessarily saying, why don't we put this in germline tissue and see whether we can connect some correct congenital condition or something like that?
Oh.
Are people likely to try that?
Well, I think the answer is yes, Eric. So that's getting more into the genome editing area, right?
Yeah.
So can we edit a stem cell and allow it to do its job? So that proof of principle has been done in vitro fertilization embryos, the ones that start to make ES cells. There's at least one publication where you can edit a gene in one of those cells to correct, I think it was one of the muscular dystrophies. And so that proof of principle has been done in a dish. Whether that will, how long, or whether that'll get into people remains to be seen. Of course, the worry is if you start editing to correct diseases in a dish, people are going to start editing to enhance various properties, and it not surprisingly, becomes a slippery slope. And certainly, just like when embryonic stem cells came out, it took a lot of thought to come up with the National Academy's guidelines to deal with this. I think gene editing, we're going to see a lot of thought in how to do this in a safe, ethical fashion. Because fixing a disease mutation in a stem cell in an IVF embryo is one thing, but those boundaries are going to get blurred quickly if we don't have some guidelines in place. Mm-hmm.
Go ahead.
You showed a map with indicating the 6 states that outlaw stem cell research,
Mm-hmm.
followed by a map that showed the distribution of the original cells, I think you said.
Mm-hmm.
And if Michigan outlawed the research, what happened to the stem cells?
So that was, I should have prefaced that map was in 2006. And so maps, like politics, can change from year to year. So that is no longer the current map of things. So some of those states, such as Michigan, have changed their policy. And actually, the University of Michigan has a large stem cell research presence. So, like the U.S. policy changed from Bush to Obama, some of the states have changed their policies. And right now, I think there are only 3 or 4 that formally outlaw it.
If not, thank you very much.